sketch out a very simplified version of the citric acid cycle. We're going to start with our input molecule, which is the acetyl-CoA. Remember, this acetyl-CoA is coming from step 2. The acetyl-CoA is processed pyruvate. So the acetyl-CoA is going to enter into this cycle, and because it's a cycle, I can go ahead and write out that refresh looking button. We know we're going to wind up pretty much back where we started. So we need something to join the acetyl-CoA in order to get citrate. I like to use the name citric acid cycle because I know one of the molecules in it is citrate, and that helps me remember to include it. But acetyl-CoA by itself is not just going to become citrate. It needs help from another molecule, and it's called oxaloacetate. So the oxaloacetate and the acetyl-CoA interact with one another, and your result is citrate. There's a lot going on here with intermediate molecules, but we're not going to worry about that for now. We can just simply write that there is a lot of enzyme modification that takes place. So much so that we modify citrate enough that our citrate molecule is turned back into oxaloacetate and is recycled. Another thing I want you to remember here is that from one molecule of glucose, we get 2 molecules of pyruvate. And from each of those pyruvate molecules, we get an acetyl-CoA. So I'm going to draw in a second one. So there's my other molecule of acetyl-CoA coming in, combining with another molecule of oxaloacetate to become citrate, which is then modified extensively by enzymes to be recycled back into oxaloacetate. So you can see now that I have both acetyl-CoAs from step 2, and that's going to help me when I'm figuring out the products that we have. So let's talk about what we get out of the citric acid cycle. As we're modifying citrate with lots of enzymes, we get some very important molecules out. We get 3 molecules of NADH, because 3 NAD+ molecules have accepted an electron and a hydrogen that goes with it. We also get another type of electron carrier called FADH2. That one's extra nice because now we picked up 2 hydrogens. We get 1 ATP, so an ADP has picked up a phosphate somewhere here. And we get 2 molecules of our waste gas, which again, remembering the equation, is CO2. So 1 molecule of acetyl-CoA, so I'm going to say the black square here, when modified before it is recycled into oxaloacetate, we get a lot out of it. We get 3 molecules of NADH and 1 molecule of FADH2. These are electron carriers and will be important in step 4, which will be in the next video. In addition to those electron carriers NADH and FADH2, we get 1 molecule of ATP and 2 molecules of waste CO2. However, we've had 2 molecules of acetyl-CoA enter the citric acid cycle. So we have a whole nother molecule that joins with oxaloacetate to become citrate and is processed by lots of enzymes. So in addition to all of this that we got, we also need to reflect that we have 2 molecules of acetyl-CoA that have been modified. So in this one, we get another 3 molecules of NADH. We get another molecule of FADH2. We get another molecule of ATP. And we get another 2 waste gas CO2 molecules. Because we go around the cycle twice, we essentially double the outputs. And we go around the cycle twice because we have 2 acetyl-CoAs. So let's get the total. Well, we just have to add. You can add straight across. I'm going to put like a total column here. We have 6 NADH. We have 2 molecules of FADH2. Remembering that both of these are electron carriers we're going to need later. We have 2 molecules of ATP. That's some usable energy, but it's not nearly all of the ATP we get from cell respiration. And last but not least, we have 4 molecules of CO2. These are your total numbers of molecules, and these are your outputs. So these outputs will be featured in the next video, where we talk about the final step of cell respiration, and that is electron transport.